right. First of all, I must thank Prof. Moon for accepting our kind invitation to IMMB International webinar. So thank you for coming and accepting. And we are very honored to host you today. So I'd like to introduce Dr. Moon. Dr. Hyo-Bang Moon is a professor at the Department of Marine Science and Convergent Engineering in Hanyang University, South Korea. He is an environmental analytical chemist with good knowledge in environmental chemistry, wildlife ecotoxicology, and human exposure to persistent organic pollutants, or the abbreviation is POP, and also emerging contaminants. He got a PhD degree from Pyongyang National University in 2003. And in the past, he had worked for the National Institute of Fisheries Sciences as a research scientist. Since 1999 until now, he has published over 230 peer-reviewed articles, which are related to marine pollution, bioaccumulation, biomonitoring, and human exposure to the contaminant, as well as the emerging contaminants. So his current research includes an investigation on environmental fate and bioaccumulation process of the persistent organic pollutants in coastal area, coastal waters, top predatory wildlife species such as shark, marine mammals, and birds, as well as humans. And he was selected as a review committee under the Stockholm Convention of the United Nations Environmental Programme during 2020 until 2024. And he is also serving as associate editors in Ecotoxicology and Environmental Safety, as well as Frontier in Marine Science, Marine Pollution. And he also editorial board member of Chemosphere, Bulletin of Environmental Contamination and Toxicology, a Journal of Emerging Contaminants, and several domestic journals. So with that impressive biography, I would like to invite Dr. Moon to share his talk with all of us today. And for the participants, if you have any question, you can always post your question in the chat box, or later on, you can switch on your mic and you can ask directly your question to Dr. Moon. Okay, all right. So Dr. Moon, the floor is yours.
Okay, thank you very much, Hyo, for your kind introduction about me. So it is my pleasure to be a speaker today in your institute and your university. So Hyo and I met in a Korean Antarctic station named Sejong through the Chile. At that time, we made this many in-depth discussion about the environmental toxicology and the microbial degradation. Unfortunately, we got some of the COVID-19, so we did not proceed more about our further studies. So I think this is a good time to start about our new study or the new project. So as mentioned by him, so okay, let me check some here. Some can you see my presentation?
Not yet.
Not yet. Okay. Sorry. Yeah.
Yeah. Now we can see your slide. Yeah, you can make the slideshow.
Okay.
All right.
Okay. Yes. Yeah. So mentioned by here also, if you have any question during my presentation, leave your message on the chatting board. I will make my answer to you. So today, I'm going to talk about the placental transfer of the polyfluoroalkyl substances, PFAS, in marine mammals. So my research background is mentioned by here, so the environmental analytical chemistry. Among them, so I have focused on bioaccumulation processes of the emerging contaminant in top predatory species from marine ecosystem. So among them, the marine mammals, including whales, dolphins, sometimes seals, finny is very valuable species to represent how the chemicals affect to our ecosystem, even ultimately to humans. So today I'm going to talk about the placental transfer. It's a very important issue in our scientific society. Okay, to introduce myself, so I made the word cloud after putting together all the title of my publication during the recent years. So you can see here some the Korea as a Baltic. So it means that I'm the expert on Korean environment. And also my research background is based on the analysis for contaminant, health, waters, exposed, persistent. So I believe you understand what I did and what I'm doing about my research. So go back to the school. So my research background is environmental analytical chemistry. So as you may know, the Earth's environment consists of the four spheres. So you call the natural ecosystem: the atmosphere, geosphere, biosphere, hydrosphere. So in the first time after the Earth system was occurred, they made the good relationship or exchange systems without any waste or unwanted byproduct. However, among them, one sphere was joined, and they made many different aspects. We call the ancestral or sometimes the society, anthroposphere or ancestral. Yes, man made many unwanted byproducts for ecosystem. Among them, I'd like to focus on the toxic chemicals. Yeah, so how we are affecting to marine ecosystem from the use of the chemicals. This is my issue. Even to Antarctic region, sometimes very deep sea organism, sometimes directly to human. So as you may know, we have too many chemicals. I don't know why. This is just a year. You are now using too many chemicals. So I believe you know about the chemical abstract service registry number. The CAS number is the unique identifier, yeah, for the registered chemicals for industrial purposes. So if you want to buy certain chemicals, you do not need to full name of the chemical. You can just suggest the CAS registry number, which means the CAS registry number is a good indicator how you are now using or consuming the chemicals. Okay, 10 years ago, yeah, almost 2010 here, so we used 500,000. Now, so in 2015, we are now using or registering over 100 million chemicals. Right now, I believe some 150 chemicals. So based on this, the time trend of the chemical numbers, we are now registering or producing 5,000 chemicals per day. Yeah, 5,000 chemicals per day. This is real. So I'm not sure all kind of every year's chemicals are safe or not. So many people, many environmental chemistry, environmental toxicologists are concerning on the safety of these newly introduced chemicals to industrial market. So too many chemicals are exposing to humans and wildlife. Among them, so fortunately, almost all chemicals could be degraded under the environmental processes as well as the biotransformation processes. Yeah, so you are lucky. However, certain chemicals could not be degraded in the environment and biological system. Those kinds of chemicals we call the persistent organic pollutant. Yeah, we call the PFAS. Yeah, as you may know, some of the K-pop is very, very famous in many countries. So the persistent organic pollutant has the four environmental characteristics. The first one is the persistence, bioaccumulation, long-range transportability, as well as toxicity. Okay, let me imagine some of the chemicals transport. So if any chemicals are remained in their source region, like in Korea, in Malaysia, like that, maybe the chemical contamination is just a local issue. However, if the chemicals are migrating, to some of the remote areas, even to Arctic or Antarctic regions, the chemicals should be controlled by global regulation. So we call the PBLT. If any chemical has properties with PBLT, the chemicals can be designated as persistent organic pollutant. United Nations, especially United Nations Environment Program, listed the 12 chemicals as persistent organic pollutant in 2001. So dioxin, furans, polychlorinated biphenyls, the DDT, and any other organochlorine pesticide. After 2009, newly added PFAS chemicals have been listed under the Stockholm Convention. Yes, the long-range transportable chemicals are not the local contaminant. This chemical should be treated as a global pollutant. So among them, today I'm gonna talk about, I'm gonna focus on perfluorinated alkyl substances, PFAS chemicals. We have many data on individual chemical groups among PFAS. However, PFAS is very high concern as well as a global concern because this is a big issue now in our scientific society. Yes, PFAS are very persistent, very stable from environmental degradation processes. So they are remained for a longer time in the environment as well as ecosystem. So we obtained those kinds of marine mammal species, common dolphins, Finless porpoises, as well as minke whales. We found the DDT and PCB show the highest concentration of any other PFAS chemicals in marine mammal tissues. This is a very interesting story because DDT, PCB, we already recognize those kinds of chemicals are very toxic, and this chemical disrupt our ecosystem, our natural biological system, as well as a natural ecosystem. So Korean environment, as well as all kinds of countries already declared to eliminate or to stop the production and use of the DDT and PCBs 50 years ago, 60 years ago. Those kinds of species were boycotted in almost 10 years ago. However, even though DDT and PCB were completely banned in industrial market or agricultural market, but those kinds of chemicals were detected or they are detecting in top predatory species from marine ecosystem, which means that they are not biodegraded, they are not degraded, they are not eliminated, they are very persistent. So they can make many undesired effects to ecosystem as well as humans. Yes, you have evidences on the bioaccumulation of those kinds of chemicals in Korean women. Almost the same pattern. So marine mammal, we analyze some of the PFAS chemicals in Korean women living in Daegu, which is the central part of Korea. So we found still high levels of PCB and DDTs in Korean women's adipose tissue because we, humans, marine mammals are sharing fish and shellfish as major diet. Seafood is the major exposure route of the PFAS chemicals to humans as well as marine mammal species. So anyway, so it's very surprising. PCB, DDT completely banned 6 years ago. However, they are still detected in our ecosystem as well as a human. So let me back to today's topic. So why marine mammals for biomonitoring? Yes, there are many reasons why I choose some whales or dolphins for my study, as well as sometimes finifed. Among them, the marine mammals can indicate the state of the food web. So you can see, so marine ecosystem and human society, there's no any relationship. However, I don't think so. Okay, you can see about some the food web structure. So marine mammals are here. It means they are top predator species. The marine mammals are healthy. Maybe their diet, like the fish and shellfish, should be healthy. Okay, how? Some of the fish and shellfish as a major diet of marine mammals can be healthy. Some of the seawater should be healthy. The seawater's contamination defined as from land-based contaminant. Okay, so land-based contaminant are producing by human. Okay, so marine mammals' health condition is directly related with humans' health. So many people now, so especially human exposure human studies, many people are recognizing some of diseases are from chemical exposure. We call them some environmental disease. So marine mammal can indicate some of the food web health system as well as some human health. Because and also so the marine mammal is very similar to the lifespan with the human, and also they can show us some the long-term chemical exposure like humans. So we choose as marine mammals for our study. Okay, let me introduce the three cases about the relationship between POP exposure and marine mammals. It was published in 1998 in big journals, in Nature. The title was "Do Flame Retardant Threaten Ocean Life?" It was published by Jacob Dubois, who is the editor-in-chief of the Chemosphere. So at that time, the PBDE, PBB were the emerging contaminant. Now many laboratories can analyze those kinds of chemicals using the instrument. However, it was the first finding in the sperm whales. It means some of the sperm whale is a kind of the deep sea organisms, which means that they can eat very cleaned seafood like some of the squid. However, PBD, PBBs were detected in sperm whales living in the central part of Pacific Ocean, which means PBD and PBBs are ubiquitous contaminant around the world. So this is just a finding. However, it was very impactful at that time because the PBDs were used for electronics and electrical equipment as a flame retardant. Flame retardant were detected in marine mammal species in 1999. It was a surprising news, and then it was published in Nature. After 10 years, the relationship paper between PFAS exposed and marine mammals. The group is Kelly and Economo, who is the Environmental Canada. So they they found some the one factors. Before this year, many people are thinking about those kinds of the marine biomagnification of PFAS are dependent on food web. For example, PCB 153 is always the biomagnified. With increasing trophic level, the PCB concentration increased. It means they showed some significant correlations between both factors. It means increasing trophic level, like top predator species, can take higher levels of PFAS chemicals. However, if the food web are different, like this one, PC-based food web, beta HCH could not be biomagnified. At that time, we believe so KOW value, the partitioning coefficient of octanol and water, was very important. It's very crucial to decide the bioaccumulation process of PFAS. However, when you consider marine mammal, you have to consider about another factors, KOA, octanol, and air partitioning coefficient, because the marine mammals can freeze like a human. This is what's new finding observed in 2007. After 10 years later, so one paper was published in Science again. This group is specialized for killer whale population as well as chemical pollutions. The title was Predicting Global Killer Whale Population Collapse from PCB Pollution. yeah, they constructed some prediction models between the killer whale population and the PCB exposure. The PCBs can make some the undesired effect on reproduction and immune function. And they found some the 100 years later, killer whale populations could be completely destroyed or some collapsed, which means some the PCB. It's just used 50 years ago. However, they can make the ecosystem destroy as the major factor. So it's very, I think I could not accept this one still because the PCB is completely banned 50 years ago. Why? Why? Yeah, because this is very persistent and very toxic. So if you decide the use of any chemicals, you should consider about their safety, in the longer time, So if necessary, we should include all kinds of technology, to confirm the safety as well as toxicity of the chemicals. So I think some of the marine mammal can say us so many things, especially some of the chemical exposed and their populations. So our concerns that the human is safe still. So today I'm going to talk about some focus on the per and polyfluoroalkyl substances, PFAS. It's very unique chemicals. Basically this is a very stable chemical. They are the bond. The CF bond is the stronger than the CCl, like a dioxin, which means that they are very stable. And also they are repellents. They have some very unique property like repellent to both water and oil. So they can be used for the surfactant. They can behave like a surfactant. So they can be used for outdoor jacket and also some the frying pan and also some the single-use food. The packing material. So and also some the very specialized like some the high-tech industry like a semiconductor aging, the cleansing agent, like those kinds of some the usage can make some the unwanted or the PFAS contamination in ecosystem as well as bioaccumulation. So I'm not sure you know about this film, the Dark Waters. He is some the Mark Ruffalo. This is based on the fact. Now still the PFAS issue is an ongoing story in the United States. So especially this is some the one big company, chemical company. Now some 3M and DuPont company produce the PFAS and PFAS chemicals. So now still many people are suffering from PFAS exposure, through the drinking water, of the groundwater. So now you can find out many some the PFAS related news in New York Times, drinking water contamination in towns of New York. So many people are suffering from the PFAS exposure. They can make some the cancer, especially the breast cancer, and sometimes some the handicapped people was some the born in some of the specialized the town. So many some the legal suit is ongoing about the people's environmental problem. If you have time, please watch some of the Dark Waters. Through some Netflix maybe. Okay, so let me introduce some of the simply environmental fate of PFAS. Okay, yes, all kinds of chemicals are derived from the terrestrial environment. We call the source to ocean. Through some the atmospheric deposition and sometimes the discharge of wastewater treatment plant. And sometimes the waste landfill can reach some of the chemicals into the marine ecosystem. And then based on the food web structure, the chemical concentration amplified through food chain, with the elevated concentration, especially top predator species can show a very high level of the toxic chemicals. Sometimes some the killer whale or other top predator like this species, human can take some of the higher position in the food web. So in this study, we used some of the Finless porpoise species, which is the distributed along the Korean coast. they use some of the shrimp and squid and other crustacean crabs as a major diet. And there the big the size is less than 2 meters. It's very relatively the small sizes. So we analyzed different types of the chemicals. Our first trial to analyze the chemical was conducted in 2015. We obtained this species from the western part of Korea, and then we transported these species to Station Research Institute in Ulsan, eastern part of Korea, and the animal doctors joined to dissect all some the species, and then we made some the different 4 groups: immature males, immature females, mature male, and mature females. And we obtained some of the biological information to interpret our analyzed data. Okay, simply I'd like to introduce some of our findings here. So we found the DDT PCB. It was obtained in 2015. Still DDT. I think DDT PCB is a never-ending forever chemicals in our ecosystem. We just should wait for completely ban in our environment. Only problem, they can make many undesired effects to our ecosystem as well as human. Unlike some PFAS, the legacy PFAS groups were detected with high concentration in the liver samples. They are relatively bioaccumulated in protein and tissues. Okay, we found very interesting findings. For example, like this, we compared immature male and immature female here, red color, and green color mature male. So significantly higher concentration of the all kind of chemicals, in the mature male. It means age-dependent accumulation. With increasing body size, the chemical concentration increased. So after birth, the chemical concentration increased with increasing age or increasing body weight. It means they are age-dependent chemicals. However, we add some mature female species. Female species concentration relatively lower than was found in mature male, as well as sometimes mature immature males, babies. It means some female species could have elimination processes of those kinds of chemical groups to their babies. So after getting on these findings from our experimental study, and we made some new experimental design. I will explain about that. And also we simply evaluated our findings compared to some of the threshold values, PCB, DDT, PFAS. Our findings, our measured concentration in marine mammals exceeded some threshold values. So threshold value were associated with endocrine disruption or immunotoxicity. Still, I do not believe this one. So PCB, DDT completely banned, 6 years ago. However, right now, still the chemicals are alive in our ecosystem, and then bioaccumulated, biomagnified in the food chain, and then they can affect to our species populations. So immunotoxicity means that they can take the disease very easily. And endocrine disruption, this is the basic metabolic activity. They can make undesired effect to our ecosystem species. So we can move our thought to humans. Maybe you are not safe from the chemical exposure. So obtaining this data, we made some new study design, like some body distribution. Sorry about some of the Korean news. So after when you do some dissection work, we found those kinds of babies of Finless porpoises, and then we made some of the new experimental design, like the transfer rate to calculation of the transfer rate of PFAS from mothers to their babies. Okay, you can imagine how some of the chemicals are transported from mothers to their babies. So major route is those kinds of the cord blood. Yeah, cord. Yeah. So when you do study about the maternal transfer can be divided into two ways. First one is a placental transfer. Second one is the other one is the breast milk consumption. So we focused on the placental transfer. However, all kind of chemicals cannot pass through placenta. It means the mothers cannot give all chemicals to their babies because the placenta has the big barrier. This is like a membrane, selective transport. It means some chemicals can transport through placenta, but some chemicals cannot be. Yeah, some chemicals blocked. Yeah, to protect some of their babies. So if you do regulate, if we design some the regulation program, what chemicals should be prioritized? I think the placental transfer chemicals should be prioritized because they can affect to the next generation. Babies are not infants are not small adult. The baby's infant should increase all organs like brain. But during this stage, the chemical exposure is very crucial. They can make undesired symptoms. Sometimes the brain could not be increased. Yeah, maybe their IQ, yeah, it's very bad. We think we should consider about some of the placental transfer chemicals should be concerned on our regulation program. To make this, we conducted some analysis. Yeah, before our this study, already our research group finding some good relationships, cord blood and maternal blood, some significant correlations with the placenta in human biomonitoring studies. Yeah, so see, Dr. Chung now see is the postdoc in the Belgium. She found some good significant relationship between the placenta and cord and maternal blood. It means the placenta concentration for chemicals and where the equilibrate with others, the mother's blood or the cord blood. So we want to apply this finding to the marine mammal populations. Okay, so what is some the basic metabolism? If the chemicals exposed to our body, the chemicals can be detoxified in our liver system. After detoxified, the chemicals can be moved to blood circulation, and they may, if the chemicals meet some places could be the good repository tissues, repository organ based on their the physical chemical properties. Yeah, we can make some good hypotheses of how the chemicals are circulating or the transport in our body, as well as sometimes they could be some excreted from our body. This is a normal metabolic system. However, after so the women or the female species could have some babies, their all metabolic system is completely changed. Yeah, because they can the placenta barrier could be active as some of the big barriers, and also some of their babies also starting some of their metabolism. So like we call the heart species can undesired some metabolic capacity. So this is a good example. Here is some of the immature species for common dolphins, mature species. They can show us a very similar PCB distribution. However, here mature female species, okay, the compositional profile is completely different. Why? I think some mature female and mature male are exposed to for the same diet like fish and shellfish, but the PCB composition is completely different. This different composition profiles is originated from different metabolic capacity. After reproduction, after pregnancy, the female species body can be completely changed in terms of the chemical exposure. So to confirm this, we obtained some we called some the failed samples, the mother fetus, the pregnant species, the 4 groups. Okay, so limit the time that I will start. I will go fast. And then we analyze the PFAS chemicals. This is okay. This figure looks very busy. Some the liver, kidney, and very hard different tissues or organ. And we found some with increasing fertility. Fertility is the numbers of the reproduction. If the reproduction number increase, the chemical concentration decline. Yeah, this is very interesting. It means the first baby could take higher levels of concentration. Yeah, and then the next, the concentration is declined. And also the chemical composition were dependent on the numbers of fertility. For example, the increasing numbers of the fertility, sky blue color proportion declined, which means those kinds of chemicals selectively, preferentially transported from mothers to their babies. Okay, for corresponding, this figure, we measured the babies' organs for PFAS. Unlike mothers, the baby concentration increased with increasing gestational age. It means after pregnancy, the time is increasing. The chemical exposure rate is also increased. It was a very surprising news. So normally, we think the chemical the exposure level are maybe after 6 years or 7 years. No, it was a very gradual increasing pattern for all kinds of the chemicals, as well as all the tissues or organ. And also we found very similar the different composition profiles with increasing gestational age. Yes, this is the fact. So interesting. So the brain, yes, the central part of this figure, can show us a slightly different pattern because the brain has also the barriers for the chemical transport. So based on this finding, we calculated the code to placental ratio of PFAS, and then we finally calculated placental transfer of PFAS based on the fetal body burden to the maternal body burden. So with increasing gestation age, the chemical transfer rate increased. So normally the Finless porpoises have almost 10 months of pregnancy time. So we are predicting the transfer rate of the PFAS from adult to mothers to their babies. And finally, we calculated those kinds of ranges of the transfer rate from Finless porpoise mothers to the babies. Yeah, some chemicals, very negligible the transfer rate. However, some toxic chemicals, 87% of the PFAS chemicals are transported from mothers to their babies through placenta. It was very surprising. So normally the legacy parts like PCB and DDT, placental transfer rate is less than 10%. However, PFAS is almost all chemicals, especially for carboxylate. Almost all chemicals can be transported from mother to their babies by through placenta. Okay, which chemical should be prioritized? For our regulation program. I think the next generation affecting chemical should be prioritized. Okay, so in summary, the PFAS are still found at a higher concentration in marine mammals with ecotoxicological concerns, and the PFAS accumulation are associated with age and sex. Age-dependent or sex-dependent accumulation were found. So when you do biomonitoring program, so you should consider about their age or sex. If you just measure the chemicals, this is meaningless. If you want to make reasonable biomonitoring program, so you should their physiological parameters. And the fetal exposure to PFAS was related with the gestational period and the fertility of the mothers. is a very crucial factor. Yes, so maybe mothers has a good elimination processes. Yeah. However, this is a major exposure route for babies. Okay, placental transport of PFAS and their alternatives were firstly calculated, and I'd like to emphasize that PFAS should be eliminated from our surrounding environment to protect us, our next generation population as well as the next generation babies. Okay, so even though we did not do any membership training from my laboratory, we made a small healing camp. My laboratory name is a Human Ecology Analytical Laboratory. So we call the healing camp. It was conducted some 2 years ago. It was the last time. Next month we will have again some the new membership training of my laboratory with some solving the COVID-19 issue in Korea. Okay, thank you very much for your listening.
Thank you very much, Prof. Moon, for your presentation. Yeah, yeah, it's a great presentation.
Thank you.
Thank you so much for making this understandable. Yeah, for many of us here who are not chemists, but we can understand your presentation content well. All right. Now we can open Q&A for Professor Moon. Yeah, if you have any question, feel free to switch on your microphone and ask directly your question. Or alternatively, you can also type in your question in the chat box. I will read it for you. All right. So there's one question from Dr. Daniel Gun. He said, Prof. Moon, wonderful talk. May I know which instrument you use to analyze the EDCS, GCMS, or LCMS?
Okay, thank you for your good question. For PFAS, perfluorinated compound should be analyzed by LC-based mass spectrometry, LC mass. Mm-hmm. However, so when you do analyzing PCB and PBDE DDT, you should employ some GC-based mass spectrometry. But however, just this is kind of the several part of the PFAS. For example, you are now analyzing some GC-based PFAS chemical groups as well. So normally the PFAS will analyzed by LC mass, but we can use some now GC mass for different property-based PFAS chemicals. Yeah, thank you very much for your question.
Okay, thanks for that. Okay, any more questions from the floor?
Oh.
Okay, so there's one more from Uma Hamza. Okay, may I know how long is the duration to study for the maternal transfer part? Do you follow from the start of pregnancy?
Okay. This is a good question. Okay, so all the studies can be designed by the research group, including me. If the sampling design is active, we do some active sampling. However, some the pregnant the marine mammal species could not be obtained with our intent. We just use some only the pregnant species from our all effort, and also we cannot catch some the marine mammals. This is illegal. So we should use some already bicoat samples in Station Research Institute with there some full support. So we cannot control some gestational age of our samples. We just use some already pregnant species. We can just check for the size of the body weight, and this is our effort before our dissection. Yeah, thank you for your question. Yeah.
All right, thanks, Uma. Thanks, Prof. Moon. All right, is there any more questions? yeah, feel free to ask. We have the expert here. All right, maybe I can ask one question to Prof. Moon.
Yes.
I have one question. So you mentioned that the DDT was found, right, at the top predators, right, the DDT. So my question is, when the top predators die, where did the DDT go? Were the DDT back to the ocean or back to the scavengers who decompose the dead body?
Yeah, this is a good question here about your research background. Yes, as I know, some top predator species are dead. They will be dropped into the underlying sediment part of the ocean. So, and then they can be consumed by some scavenger. However, they are still contained some the significant amount of the DDT or any other chemicals. Maybe they will be go back to the sedimentary environment. Yeah. So when you think about some phase out of chemicals, like we can calculate the half-life of the chemicals. For example, air samples, the soil samples. Okay, half-life is 1 hour. Maybe their concentration diminishes some the 0.5% of chemicals after 1 hour. Yes, right. This is a good definition. However, in real world, real ecosystem, the DDTs are not phased out because some of the top predator species, as that they can be exported to the new contamination into the sedimentary environment. Yes, I think some for the in the sedimentary environment, maybe they will be started with the biodegradation, microbial degradation processes, I think. Yeah. So I'm not the expert on these processes, but during this process, many chemicals can be transformed to the other form. Sometimes some chemical can be formed as a new toxic form, like methyl mercury is very important, very representative example. Just mercury can be changed into methylated mercury. This is a very toxic form rather than the ferrant mercury. Yeah. So maybe I think this is a good topic. Only problem you can reach to some the underlying sediment.
Yes, the ocean bed, right? The ocean bed. Yeah. We should follow the whale fall, the dead body of the whale and collect the sediment and analyze.
Yes.
For the
yes. If you take some indicator or trace of some biotransformed some DDTs or the other originated from marine mammal, I think it could be very good finding for us.
Yeah. I guess so. Yeah. Thanks for your answer. All right. Okay. So I also noticed that you mentioned about the PFAS in the amount of the PFAS found in the fetus, right? Is there any studies show that the newborn or the neonate has any significant change in their physiology or behavior or lead to any diseases?
Yes, definitely. Yeah, even though I'm not the expert on the epidemiological studies, many big size cohort study, like some the CDC cohort study or other countries cohort study showed some the strong or significant relationship between the people's exposure and the children's disease, like some the ADHD and other sometimes the newborn baby related disease. So I think the epidemiological studies did not support fully the cause and result, the causality between chemical exposure and their disease to cause.
Right.
However, some the strong associations have been reported for PFAS exposure to the newborn babies. So interestingly, normally all kind of chemicals for the newborn babies, the chemical level is very low. And with the increasing age for human, with the increase not the marine mammals, the babies, the exposure levels increase with age. This is very normal status for all kinds of chemical. Unlike some of the other chemicals, PFAS, the newborn babies already significantly higher than sometimes children or other adult. Because some of the placental transfer is a major exposure route for PFAS. This is why we should the concern on the PFAS exposure. So it means the newborn baby is already contaminated by PFAS from their mothers, and also they can show us many diseases. This is some or why some of the PFAS chemical should be controlled by government or the companies.
Sure. Yeah, I think so. From your data and result, they show that PFAS is really something that should be very concerning. May I know, is there any international movement to reduce the usage of PFAS? How to reduce PFAS, at a global stage?
Yeah. So as I mentioned in my presentation, PFAS is very stable chemical compared to any other chemical. For example, dioxin. Yeah, dioxin is very strongest toxic chemical. However, the PFAS is really stable. So up to now, so we did not find out any microbiology to degrade the PFAS. Yeah, this is a fact. It means they are really stable. And also, however, especially some the water treatment facility, the science part is trying to find out the suitable treatment technology, the PFAS from the water samples or drinking water. As I know, so the US EPA and the European countries are examining suitable technologies for removing the PFAS. I think, however, this is not the method for destroying the PFAS in the samples in the water. This is just based on the adsorption or absorption method. So it means some remove from PFAS from the waters. So the best way is to not use the PFAS before production. This is the best way. So we can declare, to stop the production of PFAS. And but still too many PFAS were consumed in our the society and ecosystem. Yes, we should try to find out some suitable method. Yeah, so until now, so we do not have any reasonable method.
I see. All right. Yeah. Okay. Well, there's one more question in the chat box from No Hidayah. Hi, Prof. Moon. May I know if there's any molecular study that you think can be included or have been included in the PFAS study?
Yes. Yeah, we have. If you input some keyword PFOS and PFOA in the search engine, we can find out some the molecular studies, like some the genomic studies and other molecular based toxicity studies. The PFAS were already nominated as a persistent organic pollutant, which means it can be treated as a global pollutant. To be PFAS, the PFAS should be checked for some different categories of the criterion. So among them, so toxicity is based on the molecular toxicity is the crucial part. However, not for all PFAS. Okay, I'm now saying the PFAS, this is a class name of the whole fluorinated compound. The PFOS, the carbon-based chemical, PFOS, PFOA are just the PFAS chemicals. This is compound-based regulation program from the Stockholm Convention. So the other chemical fluorinated compounds are not fully understood, in terms of some molecular exposure studies, I think. So if you have any concern on some remained chemical or emerging PFAS chemicals, so you can choose some among them. Otherwise, I can I can recommend some new chemical for your molecular study.
Yeah, that's great. Yeah, maybe I think we found several potential collaborators among the audience. Perhaps Prof. Moon, maybe you can leave later on. Yeah, maybe you can leave your email address in the chat box later, so that our audience and participant can contact you after this. There's one more question from Siti Suhana. Yeah, and she asked if the comparison between healthy and hypertension, which exposed to POP, was found to be not significantly different. Yeah. So what is your opinion on that? Like, so he's asking like your opinion, what are the important confounding factors that possibly affected the insignificant results?
Okay, I do not fully understand the question, but maybe you have some two groups for exposure studies. The first one is the toxicological studies, like using the laboratory animal. Second one is epidemiological studies. I think the hypertension could be epidemiology. I don't know. Maybe I'm not expert on this question, but I can say like the case by case until now. We did not fully accumulated the solid findings between associations, the PFAS or some other PFAS chemical groups to other certain endpoint like the hypertension. So maybe you are now generating many data set for PFAS exposure to humans or laboratory animals. So you need more time, I think. Okay, thank you anyway.
Yeah, I think so. I do agree with you. Well, could be the sample size is one of the factors that make your result not statistically significant, or there are other confounding factors. Yeah, the covariance and so on.
Yes.
All right.
Yes.
Yeah. Okay. All right. That's I think there's no more question in the chat box. Or if anyone want to ask directly, you can switch on your microphone. Yep, 3, 2, 1. No more questions. All right. Okay. So I think all right. I think that's the end of our Q&A session. And as you know, that we have 2 more minutes, and that is the end of our international webinar. So Prof Moon, once again, on behalf of the IMMB, I would like to thank Prof Moon for your time and sharing of your research with us. We highly appreciate it. I'm sure our faculty members and all participants today have learned something new and have been inspired by your sharing. So with that said, I thank all of you who are with us today. And please remember to fill out the attendance form, which is the link has been posted in our chat box. So thank you, everyone. Stay safe, take care, and I will see you again. Thank you, Prof Moon.
Thank you very much for our audience. If you have a chance, we can meet again. Thank you.
Yeah, and Prof Moon, please put in your email address in the chat box.
Okay, right now?
Yes. Yeah, thank you for coming, everyone. Yeah.
I'm done.
All right. Yeah, great presentation, Prof Moon. All right.
Okay, thank you.
Thank you. Okay, bye-bye. Take care. Bye-bye, everyone. Bye.